Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. In this lecture, I will be going over glycosaminoglycans and proteoglycans. Let's get into the details. Glycosaminoglycans, which are part of a complex molecules called proteoglycans, these are unbranched complex heteropolysaccharides. So the glycosaminoglycans are repeating disaccharide units which are present in a proteoglycan molecules. And within this repeating disaccharide unit, so one of the unit is an amino sugar and the other unit is an acido sugar. And amino sugar is usually acetylated and the acidic sugar is mostly it's an uronic acid. So arrangement of glycosaminoglycan is shown here. That is, it can be represented as XY and that XY is repeated. And ultimately that repetition, that polysaccharide, it is attached to a core protein. And that's how the glycosaminoglycan becomes part of a proteoglycan molecule, which is a bigger structure. So let's look at what is X and what is Y in a glycosaminoglycan. So X here in the glycosaminoglycan repeating sequence, that is the disaccharide, repeating disaccharide. X is an amino sugar, and as I said earlier, it is a mostly acetylated amino sugar like N-acetyl glucosamine, N-acetyl galactosamine, like that. And the Y is an acidic sugar, and mostly that acid sugar is uronic acid. So with that repetition, an amino sugar, acid sugar, amino sugar, acid sugar, same amino sugar and same acid sugar will be there. It is just XY, XY, XY. It just continues, that repetition continues. That's why these two disaccharide units, amino sugar and acid sugar, repeats hundreds of times. And then finally it will be attached with a protein called a core protein, and that entire thing will be further attached to a bigger protein molecule, and that will be proteoglycan. We are going to see that towards the end of this lecture. And this is how proteoglycans are made with the help of glycosaminoglycans, and the glycosaminoglycans further, these are repeating disaccharide. Let me just write down the simplified representation. So we have a glycosaminoglycans which will be attached to a core protein, and that core protein in turn it will be attached to a other molecule, usually like hyaluronic acid or something like that, with the linker protein. Okay, so that's how the proteoglycan molecule will be looking like. So there will be multiple glycosaminoglycan units which will be attached to this central molecule. So all these lines indicate several different types of glycosaminoglycan. So what is there in that glycosaminoglycan? So in this glycosaminoglycan, it is amino sugar and plus acidic sugar, acid sugar. Amino sugar and acid sugar will be n number of times. Multiple amino sugar and acid sugar will be present. Okay, and that's what is and it is unbranched. There is no branches in that glycosaminoglycan. So that's why glycosaminoglycan is referred as unbranched complex heteropolysaccharide. Alright. Now amino sugar is either N-acetyl glucosamine or N-acetyl galactosamine in most of the glycosaminoglycan molecules, except in heparin and heparan sulfate, where it is only sulfated. Now acid sugar is mostly D-glucuronic acid, except in dermaton sulfate, where it is L-ioduronic acid. Ioduronic acid is epimer of glucuronic acid. So whereas a type of glycosaminoglycan, that is keraton sulfate, it do not have an acidic sugar there. That is something exception you need to remember. Okay. So, amino sugar which is either acetylated as N-acetyl glucosamine or N-acetyl galactosamine, except in the heparin and heparan sulfate, all these are sulfated. So, amino sugar is either N-acetyl glucosamine or N-acetyl galactosamine. Acid sugar is glucuronic acid, except in the dermaton sulfate it is ioduronic acid, and the keraton sulfate do not have an acid sugar. And glycosaminoglycans are major components of extracellular matrix connective tissue, being part of a proteoglycan molecule. Now let's look at different types of glycosaminoglycans which are part of a proteoglycan molecule. So here examples of glycosaminoglycans are given. Characteristic disaccharide which is present in each of the glycosaminoglycan is given. Whether it is sulfated or not, that sulfation is given, and also tissue location is given. Very important table. Make sure you remember, understand, memorize. Make sure you know. First is hyaluronic acid. Hyaluronic acid, it is a combination of a disaccharide, that is N-acetyl glucosamine and glucuronic acid, and it is not sulfated. Hyaluronic acid is not sulfated and it is found in joint space and ocular fluids. Coming to chondroitin sulfates. As you can read here, it is chondroitin sulfates. That means there is chondroitin 4 sulfate and chondroitin 6 sulfate. Now the chondroitin 4 sulfate, where N-acetyl galactosamine 4th carbon is sulfated. In chondroitin 6 sulfate, N-acetyl galactosamine 6th carbon is sulfated. Other than that, the disaccharide unit in chondroitin sulfates is glucuronic acid and N-acetyl galactosamine with sulfation. Sulfation is over N-acetyl galactosamine here, and location is in the cartilage, tendon and bone. Keraton sulfates. Keraton sulfate as galactose. There is no glucuronic acid or ioduronic acid in keraton sulfate. So, the acid sugar is not there in keraton sulfate. It is just the galactose. No uronic acid. That's an exception. So, all glycosaminoglycans contain uronic acid, whereas keraton sulfate do not contain uronic acid. It just have a galactose. Along with the galactose, there is N-acetyl glucosamine 6-phosphate, and keraton sulfate is present, example in the cartilage and cornea. So there is keraton sulfate 1 and keraton sulfate 2 there. Dermaton sulfate. Dermaton sulfate contains ioduronic acid and N-acetyl galactosamine 4 sulfate. So sulfation will be on ioduronic acid and also N-acetyl galactosamine, and it is present in skin, heart valves and blood vessels. And coming to heparin. Heparin is, it's containing ioduronic acid and N-sulfoglucosamine. So the sulfation is on glucosamine and ioduronic acid, and it is present in mast cells. In fact, heparin is secreted by the mast cells and also present in the liver. Heparan sulfate, it is containing glucuronic acid, N-sulfoglucosamine. So the difference between heparin and heparan sulfate is the acid sugar. In heparan sulfate, it is glucuronic acid, whereas in heparin, it is ioduronic acid. So the sulfation here in heparan sulfate, it is over glucosamine. And note that heparan sulfate is attached to the plasma membrane and it is found over cell surfaces. Now these are some of the examples of glycosaminoglycans with their disaccharide combinations, that is amino sugar, acidic sugar with certain exceptions. So make sure you remember the disaccharide composition of glycosaminoglycans. So one more time, hyaluronic acid, it is glucuronic acid and N-acetyl glucosamine. Chondroitin sulfates, it is glucuronic acid, N-acetyl galactosamine with sulfation. Keratin sulfate is galactose with N-acetyl glucosamine. Dermaton sulfate, it is ioduronic acid with N-acetyl galactosamine. Heparin is ioduronic acid with glucosamine, and heparan sulfate, it is glucuronic acid with glucosamine. All of them are sulfated except that hyaluronic acid, there is no sulfation there. Now let's move on to see classification of glycosaminoglycans. So they are classified into 6 types based on their charge and sulfation. So glycosaminoglycans are also referred as mucopolysaccharides. That's why it is written as MPS here. MPS means mucopolysaccharide. Now there is a neutral mucopolysaccharide and acidic mucopolysaccharide. And among the acidic mucopolysaccharide means acidic glycosaminoglycans, there are sulfate-free glycosaminoglycan and sulfate-containing glycosaminoglycan. Majority of the glycosaminoglycans are sulfate-containing except hyaluronic acid, which is sulfate-free. Remember that. So now let's look at acidic sulfate-free mucopolysaccharide. In that we have example hyaluronic acid. So in the hyaluronic acid, the repeating disaccharide, as we have seen in the previous table, it is glucuronic acid and N-acetyl glucosamine. So here is the structure of hyaluronic acid. So glucuronic acid and N-acetyl glucosamine, it is making a beta-1,3 linkase. So this is the beta-1,3 linkase. Combination is glucuronic acid and N-acetyl glucosamine. Now let's move on to see details on hyaluronic acid. So hyaluronic acid is a special glycosaminoglycan compared to other glycosaminoglycans because it do not have sulfate groups. It is not being covalently attached to a protein molecule when it is becoming part of a proteoglycan, and it is present both in animals and bacteria, and its polysaccharide chain is the longest of all glycosaminoglycans. It has got viscous gel-like substance. It is found in synovial fluid of joints, vitreous humor of the eye, umbilical cord, and also loose connective tissue, and it primarily serves as a lubricant and shock absorbers. These are all the special functions and also characteristics of hyaluronic acid, which is special when compared to all other glycosaminoglycans. Coming to hyaluronidase. Hyaluronidase, it is an enzyme that will depolymerize hyaluronic acid, and it is present in tissues like testis, spleen, as well as pneumococci and hemolytic streptococci releases hyaluronidase. So hyaluronidase, it helps in spreading of infection or helps in the spreading process because it is going to depolymerize hyaluronic acid in the extracellular matrix. So hyaluronidase, it acts as a spreading factor by causing depolymerization of hyaluronic acid, and that's how having hyaluronidase, it helps in the fertilization process, especially coming from the sperms. Coming to acid sulfate free MPS, one more here is chondroitin. The chondroitin, it is found in cornea, has been isolated from cranial cartilages. It differs from hyaluronic acid only that it contains N-acetyl galactosamine instead of N-acetyl glucosamine. That's the only difference between hyaluronic acid and chondroitin. So it's a sulfate-free glycosaminoglycan. So in that sense, we have two sulfate-free glycosaminoglycans. One is hyaluronic acid and other is chondroitin. Now let's move on to see sulfate-containing acid mucopolysaccharides. In that, the first one is keratin sulfate. Look at the keratin sulfate structure here. It is galactose, that is making a beta-1,4 linkase with N-acetyl glucosamine 6-phosphate. So note that keraton sulfate do not contain uronic acid, no glucuronic acid or no ioduronic acid. So it is an exception here. All glycosaminoglycans contain uronic acid except keraton sulfate, which is not acidic sugar. So some of the characteristics of keraton sulfate here, next slide. It is the most unusual of all glycosaminoglycans. It is the only glycosaminoglycan that does not contain acid sugar because there is no uronic acid there. And the repeating disaccharide unit consists of N-acetyl glucosamine and galactose, remember that. And it is linked to a protein by rather unusual linkage compared to all other glycosaminoglycan, that N-glycosidic linkage with asparagine residue that are usually found in glycoproteins. So unlike other glycosaminoglycans which are attached to proteoglycan molecules, so here keraton sulfate, it is linked with the core protein with N-glycosidic linkage with the asparagine, amide nitrogen of asparagine. This type of N linkages are usually found in glycoproteins and very rare in proteoglycans. Repeat that. This type of N linkage is usually found in glycoproteins and are rare in proteoglycans. So keraton sulfate, while making proteoglycan, it is attached to a core protein with N linkage. So that's something special with keraton sulfate. Now there are two types of keraton sulfate, keraton sulfate 1 and keraton sulfate 2. Keraton sulfate 1 is found in the cornea, thereby it helps to keep cornea transparent, and N-glycosidic linkage is there with the core protein with keraton sulfate 1, type 1. That's where is the N linkage comes in, which makes it special. Keraton sulfate 2, it occurs in skeletal tissues, and the linkage here is with the O linkage, just like with any other glycosaminoglycans linking with the core protein. So here the speciality of linkage is only with the keraton sulfate type 1, which is using N linkage. Remember that point. Now coming to chondroitin 6 sulfate. Chondroitin 6 sulfate is a combination of acid sugar, that is glucuronic acid, and amino sugar, that is N-acetyl galactosamine, which is phosphorylated at the 6th carbon. That is what is chondroitin 6 sulfate is. Now chondroitin 4 sulfate is phosphorylated at the 4th carbon. So both chondroitin 4 sulfate and chondroitin 6 sulfates, they are most abundant glycosaminoglycans in the body. Remember that point. And the repeating disaccharide units in chondroitin sulfates are glucuronic acid, and N-acetyl galactosamine is sulfated on either C4 carbon or C6 carbon to make chondroitin 4 sulfate and chondroitin 6 sulfate respectively. Coming to dermaton sulfate. Dermaton sulfate is composed of ioduronic acid and N-acetyl galactosamine 4 sulfate. So some of the details on dermaton sulfate is the same chondroitin 4 sulfate is converted into dermaton sulfate after epimerization of D-glucuronic acid into L-ioduronic acid. Because the difference between chondroitin 4 sulfate and dermaton sulfate is the acidic sugar in chondroitin 4 sulfate is glucuronic acid, whereas the acidic sugar in dermaton sulfate is ioduronic acid. Let me show you the difference with the figures. So as you see here, the chondroitin 4 or 6 sulfate, it has got acidic sugar, that is glucuronic acid. So glucuronic acid with N-acetyl galactosamine, which is sulfated at 4th carbon or the 6th carbon. Now the dermaton sulfate, as you see here, instead of glucuronic acid in chondroitin sulfate, so here it is ioduronic acid. Other than that, so the other amino sugar is same, that is N-acetyl galactosamine. So the difference between a chondroitin sulfate and dermaton sulfate is only the acidic sugar, that is glucuronic acid in chondroitin 4-sulfate is now epimerized into ioduronic acid in dermaton sulfate. Other than that, the amino sugar is N-acetyl-galactosamine, which is sulfated at 4th carbon. It is same between chondroitin 4-sulfate and dermaton sulfate. So that is what is being explained here in this slide. Now, repeating disaccharide unit here with that epimerization process, it is ioduronic acid and N-acetyl-galactosamine 4-sulfate. And dermaton sulfate is present in the skin, that's why it is referred as dermaton, and blood vessels and heart walls. And also dermaton sulfate, it has got weak anticoagulant property, and hence sometimes it is called as beta heparin. So because of its weak anticoagulant property, dermaton sulfate is also referred as beta heparin. Now let's move on to see heparin. Heparin is a sulfated glucosamine with sulfated ioduronic acid. So it is a glucosamine, it's an amino sugar, and also acidic sugar is ioduronic acid. So it is heavily sulfated because both the monosaccharide units in this repeating disaccharide is sulfated. Both glucosamine is sulfated and ioduronic acid is also sulfated. Now the characteristics of heparin is, it is a repeating disaccharide of glucosamine and ioduronic acid. It is richly sulfated, heavily sulfated. Sulfate groups are found in almost all glucosamine residues. On an average, around 2.5 sulfate groups are present per disaccharide units seen. This is one of the most heavily sulfated glycosaminoglycan. It is strongly acidic due to rich sulfates, that is SO4-, and unlike other glycosaminoglycan, which are all extracellular, heparin is an intracellular glycosaminoglycan. And the core protein of heparin proteoglycan is rich in serine and glycine. That is another thing that we need to remember with heparin. These are some of the special points related with heparin. Uronic acid with few sulfates and some glucosamines may be acetylated. So it has a negligible anticoagulant activity, although the subunit combinations are similar, and also unlike heparin, this is an extracellular glycosaminoglycan and usually found in the basement membrane of kidney and basement membrane of stratified squamous epithelium, and also it is an essential component of cell surfaces, and this will help in anchoring certain proteins over the cell surface. Example, lipoprotein lipase, it is held to the membrane of endothelium of the capillaries. So these are some of the points with different types of glycosaminoglycans. So with disaccharide combination and also tissue location. Coming to formation of proteoglycans. Glycosaminoglycans interact with core proteins to become a proteoglycan molecule, and the carbohydrate content in proteoglycan is much higher than glycoproteins. Generally, up to 95% of glycosaminoglycan that is attached to a core protein, it will make a proteoglycan molecule. It is the carbohydrate because it is a heteropolysaccharide. Linking of glycosaminoglycan to a core protein occurs by core trisaccharide, and the core trisaccharide is galactose-xylose, which is also referred as a link trisaccharide. So here the glycosaminoglycan is attached with the core link trisaccharide or core trisaccharide, and that core trisaccharide is a link trisaccharide, that is it has galactose and xylose, and the xylose is the one that interacts with either serine or threonine in O-linked glycosaminoglycans to a core protein, or it can be asparagine, amide nitrogen of asparagine in N-linked glycosaminoglycans. And example for that we have seen is keratin sulfate 1. An exception here to all this type of linkage is hyaluronic acid, which is the longest glycosaminoglycan, and it is not attached to a core protein. Remember that exception. So here is the proteoglycan monomer and proteoglycan aggregates figure shown. So the core protein, it is linking a glycosaminoglycan, as you see here, this part. Let me highlight. So here is the repeating disaccharide containing glycosaminoglycan, and it is attached to a core protein. And this is one of the unit of proteoglycan molecule. So core protein is shown here, this particular part. You see the center molecule is a core protein, and to that a repeating disaccharide unit glycosaminoglycan is attached. So the in proteoglycan aggregate, the bigger molecule, all these proteoglycan monomers, they will bind with hyaluronic acid. So the central molecule is a hyaluronic acid. To that proteoglycan monomers containing a glycosaminoglycan with the core protein is attached to hyaluronic acid. So basically we have a proteoglycan aggregate, and the proteoglycan aggregate contains proteoglycan monomer, and one proteoglycan monomer has got core protein in the center, and to that core protein a repeating disaccharide containing glycosaminoglycan is attached, and that one proteoglycan monomer will interact with the hyaluronic acid. That's how the connection goes. And imagine hundreds and hundreds of proteoglycan monomers attaching to hyaluronic acid, and that makes a proteoglycan aggregate in the extracellular matrix, and the structure, entire structure, it looks like a bottle brush appearance. It is the bottle brush appearance is the similarity to this proteoglycan aggregate. So there are three types of linkages seen between glycosaminoglycan and a core protein. This type of linkages are observed in proteoglycan aggregate formation. So it's a O-glycosidic linkage which is formed between a xylose and the hydroxyl group of serine, which is found in a core protein, and this bond is unique to proteoglycan molecule. Other is N-glycosidic linkage, which are formed between N-acetylglucosamine and amide nitrogen of asparagine residue in a core protein, and typically this type of N-glycosidic linkage is found in keratin sulfate 1 and N-linked glycoproteins. So in that sense, the only glycosaminoglycan with N linkage is keratin sulfate 1. So the other type of linkage that is seen is O-glycosidic linkage, and the type of O-glycosidic linkage that is found between N-acetylglucosamine and hydroxyl group of serine and threonine found in a core protein, which is typically seen in keratin sulfate 2. Now note that the keratin sulfate is a special molecule in the sense that the keratin sulfate 1 has N-glycosidic linkage with N-acetylglucosamine, making a amide nitrogen linkage with asparagine residue, whereas keratin sulfate 2, it has a O-glycosidic linkage, but that O-glycosidic linkage is between N-acetylglucosamine and hydroxyl residue of serine and threonine, rather than linking with the xylose of linked trisaccharide, because all other O-glycosidic linkages in proteoglycan molecules, it is using linked trisaccharide, whereas O-glycosidic linkage in keratin sulfate 2 is using N-acetylglucosamine. Now coming to functions of proteoglycans molecules. Now proteoglycans are important constituents of extracellular matrix or ground substance. They act as polyanions and maintains a tissue turgor by interacting with the polycations. They act as a barrier in tissue, prevent infective agent migration, and also they prevent metastasis. And like hyaluronic acid being present in the joint space act as a lubricant. They help to release hormones. They help in cell migration in embryonic tissues. They help in glomerular filtration, especially like heparan sulfate's role there. They act as anticoagulant like heparin, which act as an anticoagulant. They can act as coenzymes, and also they act as receptors of cell, and also being present over cell membrane, they act as an anchoring molecules of certain proteins over cell membrane. I hope this lecture has helped you in understanding details on glycosaminoglycans, various types of glycosaminoglycans, composition and characteristics of each of these glycosaminoglycans, and briefly on the location of glycosaminoglycans, and overall how these glycosaminoglycans they become a part of a bigger molecule called proteoglycans. Thank you.